Geriatric Medicine Grand Rounds. Um, Happy Friday, everyone, in December. I hope you're getting safely to your destinations, driving in the dark. Uh, um, we have a really wonderful speaker today. I was just reflecting on how lucky we are to have world-renowned scientists right here at the University of Washington who could share with us their work that's really at the forefront of what we're thinking about, certainly in gerontology, but definitely beyond. So Dr. Warren Ladiges is a professor of comparative medicine in the and is the director of the Geropathology Research Network, and is also the UW Nathan Schock Center Geropathology lead of the UW Nathan Schock Center Geropathology Research. And he's really been the leader in the application of pathology and basic geroscience. You are hearing from the expert today. His research focuses on what he's going to be talking about, which is the investigation of pathological mechanisms of aging with a specific interest in preclinical testing of anti-aging drugs in aged mouse models for translational relevance to clinical aging studies and chronic diseases of older adults. He's also the editor-in-chief of the Journal of Aging Pathobiology and Therapeutics. And I really appreciate you taking the time out of your busy schedule to talk to us about something that is really at the forefront of science right now. So thank you.
Okay. Thank you, Kate. Um, so just a couple of disclosures. The work I'm talking about today was solely funded by NIH grants, especially all National Institutes on Aging. Um, so, um, so what is geroscience, and how does it relate to resilience to aging, which I'm going to talk about today? Um, so, um, it, Geroscience is really about um, pathways of aging that involve multiple aspects, and it basically defines age-related diseases as being driven by these aging pathways, so that if there are ways of preventing or slowing down aging, then by definition, we're going to be able to slow down or prevent age-related diseases. And so how does resilience fit into this? So resilience is really resilience to aging. Physical resilience to aging is really the ability um, to be resistant to various aspects of gene function, whether that's increased function or decreased function of genes involved in pathways of aging. So this combination then serves as a platform for prevention rather than treatment. And so when we talk about prevention, we need to, figure out, well, who needs, this prevention approach, especially if we're looking at middle-aged people rather than geriatric people. And there are ways of doing this that aren't fully developed yet. And the polygenic risk score using SNP chips, plasma protein biomarkers. One of the things I'm going to talk about today are physical stress tests that we're developing in the mouse model. The ability to recover from an acute stressful event. So the gerotherapeutic approach to enhance resilience to aging is really dependent on identifying individuals that are at risk for unhealthy aging and development of age-related diseases. Um, there are obviously lifestyle approaches that provide this. Exercise, diet, really low cost. I'm a firm believer in that. Um, but yeah, if the need arises, depending on risk factor assessment, then there are repurposed drugs. And I'm talking about combinations here. So um, that really will be the focus of the talk here. Uh, so this is just a, a graphic image and overview of what I just talked about. Um, Pathways of aging um, are associated with chronic age-related diseases, and we're using a number of models to in preclinical settings to look at how we might target some of these pathways of aging. And here, we're interested in, like I said, delaying the onset of aging. A lot of my colleagues have, worked with worms and flies and mice. We're, working with um, a number of mouse models, but specifically for this work, we use mice from they're all naive mice, C57 Black 6 from the NIA Agent Rodent Colony. I'm also going to mention about a new model that we're trying to develop, but we're really, doing preclinical screening of drug combinations. Um, so, um, what, what kind of drugs are we talking about? Um, so this table, I modified it from Nir Barzilai's paper published in 2024 that looked at repurposed drugs. We're really focusing on repurposed drugs because they're already approved, FDA approved. A lot of them have been around for quite a while. We know they're safe, relatively few side effects. Um, uh, and the question is, can they be combined to provide a broader scope of targeting, more pathways of aging? So I'm going to talk about three drugs today: acarbose, rapamycin, and phenylbutyrate as a cocktail. Acarbose, most of you probably are familiar with this. It's a type 2 anti-diabetic drug. Um, it's not absorbed systemically. It acts locally in the upper small intestine to prevent breakdown of complex carbohydrates. Rapamycin um, is an immunosuppressive drug that's been shown to enhance aging in a number of models and is also in a number of clinical trials. Acarbose has also been shown to extend lifespan in mice. Um, and then I added phenylbutyrate. It's a HDAC2 inhibitor, and it's a drug that we've worked with for a number of years. And so the thinking was just arbitrarily that, okay, with these three drugs, we're really going to broaden the swath that this trio should target. So um, our original study design was to test this prototype drug cocktail of rapamycin, phenylbutyrate, and acarbose. Like I said, we used both male and female C57 Black 6 mice, 20 months of age. The treatment, so these were added to the feed um, and rodent chow by period meals. We just had them integrate the drugs at the milling factory. And so the way we did this was we used the drug cocktail as one cohort compared to each individual drug and then a control. We also tested Het3 mice, this four-way cross developed by Rich Miller at Michigan. And this is the exclusive strain that's used in the intervention testing program supported by NIA. We also looked at a half a dose, and I'm going to show you some of those results. Um, so um, the cocktail consistently decreased body weight and fat mass um, without really changing food consumption. Um, and so when we look at performance, uh, we basically use three different assessment procedures. This is a special learning task. We call it a box maze. It's simply a box. Um, and as you can see here, you know, the black column is the control, the gray column is cocktail, and then rapamycin, acarbose, and phenylbutyrate. Um, so in the special learning task, both males and females responded to positively with improved learning uh, during the test time at the end of 3 months that the mice were treated. And also we showed that rapamycin performed pretty much at the same level as the cocktail. Um, uh, so if we look at the road rod, just a rotating rod, we see how long the mice can stay on this rotating rod. It's really a test of agility. Um, and we can see that in both males and females, only the cocktail had an effect. So, none of the individual drugs were effective on a significance basis. There were some trends, obviously. But that to us, it kind of suggested that must have been some additive effect. Grip strength, um, it's very similar to the hand grip strength that's used as one of the assays for frailty in older people. Um, Pretty much the same type of approach, but you can see there that again, the cocktail really improved grip strength, and yeah, and also it looks the acarbose cohort also had improved grip strength. Um, so if we look at then geropathology, and Kate mentioned the fact that we have done a lot of geropathology. We developed these lesion grading platforms in mice and rats, although we haven't published that. We're working on a platform for non-human primates as well as pet cats. So here you can see then that this really gives us a deeper inside look, representative of comorbidities, where we look at all lesions and we grade them, so we get a severity score or a severity grade. And you can see here the black column is the control, the gray column is the cocktail, um, and across every organ, the cocktail is effective in um, you know, decreased lesion severity. Um, and again, we can see our friend rapamycin, um, is almost as effective as a cocktail. So rapamycin is, as we expected, the dominant, it appears to be the dominant factor in this cocktail. Um, but so this is males. We saw a similar aspect where rapamycin wasn't quite as effective, but the cocktail was. So all major, these four major organs were responsive to the cocktail. Um, so yeah, you can see that except for in males in the kidney, where um, you know, uh, all the cohorts, um, were responsive and showed decreased lesion severity. Um, so um, I wanted to um, just kind of signal out one one organ, and that's kidney. Black 6 mice have a fairly high incidence of chronic kidney disease, and you don't have to be a board certified pathologist to see that there's a major difference here. We scored this on the right hand side as a grade 1, um, and on the left hand side is a grade 3. So this is progressive nephropathy, actually glomerular nephropathy, um, yeah, that was basically and quite robustly shut down by the cocktail. Uh, so we want to take a little closer look then, um, and did a qPCR to look at RNA message, and you can see there that the cocktail performed um, in most of the inflammatory cytokines, so that um, there was similarity to the young untreated control. Uh, okay, so um, you know what we want to do next was see if, in fact, a decreased dose uh, was going to be effective. And you can see in terms of performance, the box maze or this special learning task um, was just as effective um, as the full dose cocktail. But the other performance tests, um, the half cocktail didn't have any effect. Um, so if we look at um, then and think about the pathways um, that this cocktail might be targeting. Um, we did RNA sequencing, a shotgun approach RNA sequencing using GSEA analysis and databases from the molecular signatures database. And you can see that there was indeed, with um, basically pathways of aging, there was down regulation of a lot of um, signaling pathways that were in fact associated with pathways of aging. Um, so even down here where it wasn't quite significant, um, SAFs or autophagy, there's still um, some evidence that there was some effect. Uh, so um, then we followed up then with immunohistochemistry. I call it brain histoimaging. Um, yeah, we have a fairly large array of optimized proteins, or sorry, antibodies um, for these proteins. Um, and for digital imaging, we use the open source QPath program. Um, so you can see these heat maps. Um, the cocktail treated mice um, really tended to have heat maps that were more similar to the young untreated control. And this was, these were hippocampus sections. So, uh, and we decided, to just focus on one organ because it costs a lot of money to do RNA sequencing for a lot of um, tissues, and and so we're in the process of doing RNA sequencing in other organs from this study. So, um, so um, then what does the immunohistochemistry really tell us? Um, it tells us, um, what we had expected. That in terms of quantitative optical density readings, where the control HDAC was high, the cocktail suppressed that, um, uh, and lo and behold, as we had hoped, PBA's phenylbutyrate suppressed that. It's an HDAC2 inhibitor. Yeah, but if we look at some other markers, gamma H2X is a protein that's responsive to repair of DNA damage. And you can see that uh, the cocktail really suppresses that, suggesting that there's less DNA damage. Rapamycin was similar in its ability to do that. Um, and ATG5 is autophagy protein. And you can see it had the opposite. In other words, autophagy um, declines with increasing age, and this tended to boost that. So, um, so we have also additional um, um, data, optical density readings, showing that inflammatory cytokines, are you know, um, decreased, with the cocktail as well as rapamycin, um, and occasionally, um, some of the other acarbose and PVA, but not consistently. Um, so, and this is P21. We don't have an optimized antibody for mouse P16, so we did PCR and we got the same results here. Um, so this suggests that at least in the brain, this cocktail is actually, um, um, yeah, I'll just say it's, we think it's probably targeting these pathways. Is it targeting other things? Probably, but that may come up when we look at other organs more specifically. Um, so. We also, as I mentioned, wanted to test the cocktail in a different strain. These were what we call Het3 mice, four-way cross. Um, 1/4 of the genetic background is from C57 Black 6, by the way. Um, and you can see that body weight and fat mass were decreased by the cocktail, similar to what was was done in the Black 6 mouse. But this was a little disappointing. Absolutely no effect was seen in our special learning task. And we know that this the Het3 mice don't develop age-related cognitive decline as soon as Black 6 mice or the F1 B6 F1 hybrid. So I don't know if that's a factor or not, but again, this was an unexpected finding, and it's only effective in males on the rotating rod, um, and then on the grip strength, it had some effect as well. So that oh, and we also did uh, geropathology, and again, there was inconsistent findings in terms of what organs were actually affected. In females, the liver didn't seem to be responsive, um, and in males, the lungs. Um, so again, this was kind of unexpected, but it just shows that there's a strain dependency and may indicate that there may be a need for in screening drug screening tests. There may be a need to do two mouse strains and rather than just depend on one. Um, uh, so, um, so where are we then? We have this prototype drug cocktail, and it's obviously, uh, in general, more effective than each individual drug. Um, it seems to target pathways of aging, at least in the brain. Um, there there is definitely an additive effect of the three drugs, but there's no synergism. Um, so we feel it's a promising prototype. But the real question is who needs this? Um, and here's where I want to kind of get into um, the next section of the talk, and talk about risk assessment, because that's what we're really dealing with. We don't want to um, indiscriminately go out and treat everyone with a cocktail of rapamycin, acarbose, and phenylbutyrate that's middle age. Um, we only want to administer this or some similar cocktail on an as-needed basis. So it's more about individualized geriatric medicine. Um, but so we were fortunate enough to get a grant funded several years ago to look at physical stress tests. Um, and we did fairly well. We got a number of publications, and actually were able to get a non-competitive renewal funded recently. So we have an opportunity to do a lot more work on this. So when I say physical stress tests, I'm talking about administering something, for example, a skin biopsy. Here, it's a ear punch biopsy in the middle part of each ear in the mouse, and we look at wound closure as a readout. We can separate um, we call them fast healers versus slow healers, um, and then correlate that, after a time with various aging parameters, and I'll show you the strategy. Um, so look, these are just some of the ones we've worked with. I'm going to talk about the ear punch biopsy, the sleep disruption, and cyclophosphamide. So we'll get right into it. Um, um, so it's really about predicting resilience in middle-aged mice. Um, so each stressor was administered to black 6 mice at 18 months of age. Um, and as I said, we were able to discriminate into basically a resistant group and a susceptible group. And then at 26 months of age, we did some of the performance assays and geropathology to see if there's any correlation between the ability to predict resilience or predict non-resilience, right? Because that's really what we're looking at in terms of risk assessment. Um, so here is um, data showing correlation analysis and associations, you know, with um, memory and ability to stay on a rotating rod and grip strength. So this box maze slope, we call it a box maze because it's a box, um, you know, and yeah, it really is uh, um, a we call it a spatial navigation a task for learning. And you can see, in terms of the slope, by linear regression analysis here, that in fact, um, you know, um, fast learners, in other words, the ability to respond to sleep disruption, because, so we have this um, protocol where we're able to show by disrupting sleep patterns in mice during the middle of the day for 4 hours for 4 days, that there is rather a robust cognitive impairment. And so, we can separate these mice into, again, um, mice that are relatively unimpaired versus mice that are more severely impaired. And you can see that as expected, that the fast learners are associated with better memory um, at the end of this monitoring period, which was 26 months of age. Similar findings with the rotor rod. In other words, um, the fast learners were associated with mice that stayed on the rotating a bit longer. The, However, there's no association with grip strength. So if we look at cyclophosphamide, cyclophosphamide is an immunosuppressive drug. It targets myeloid precursor cells. Um, and if we give one dose, it's a small dose. Um, there is immediate nadir at 3 days, and I can tell you, this is so consistent that it's almost scary. Um, there is a nadir at 3 days and a rebound response at 7 days. So that time between 3 and 7 days, um, is different in each individual mouse. So we standardize the data using the neutrophil lymphocyte ratio, and you can see that in terms of staying on the rotor rod, lower NLR on day 7 compared to day 3 um, is associated with better performance on the rotating rod as well as increased grip strength, but really had no effect on memory. Um, what about then wound healing? We use this ear punch biopsy, 2 millimeter ear punch in the middle of each ear, and we can measure um, the wound closure, the rate of healing, basically. And again, separate into two different groups, fast healers and slow healers. And you can see that fast healers uh, predicted increased grip strength um, and also increased memory on the water tread maze. This then provides I should talk about, we also did geropathology in these mice and showed that, um, the response to sleep deprivation was predictive of lower lesion scores. CLS just stands for composite lesion score. Um, uh, and so, kidneys, um, uh, the lesion scores, were much lower um, in mice that were um, fast heat, or mice that responded to sleep disruption, um, and, and lung um, severity scores were lower in mice that responded to cyclophosphamide. Um, and liver lesion scores were lower in mice that were fast healers, in terms of the wound healing. Um, so, what does this tell us then? So if we kind of summarize what this really means, and again, I put this kind of on the other end where we can identify mice by these physical stress tests um, that are not resilient to aging or age-related diseases. Um, and so, sleep disruption, and we have, poor memory, um, you know, uh, decreased kidney lesions, and you can see similar, but not necessarily, overlapping uh, predictive abilities, right? Um, so we got the idea, well, let's just combine them as a panel. Um, and we're in the process of evaluating that data right now. But and also, um, the fact that these are can potentially be converted to in vitro tests. In other words, um, we're looking at we're doing RNA sequencing. We're looking at plasma proteins. Those types of things that can be a rather simple, straightforward in vitro test. Um, and so that's sort of where we're at, with these physical stressors. And again, this is only one way of risk assessment. Um, as I mentioned, there are other ways of doing that. Um, so, um, uh, so where are we currently? So, um, I think everyone would agree that it appears that the cocktail is, in fact, enhancing resilience to aging. Um, Treated mice had more useful like cognitive function. They had retained grip strength and agility. There's less evidence of chronic disease and other comorbidities. Um, and we're at the point where we can use physical stress tests um, to see if we can actually predict um, which mice might need something like this um, uh, drug cocktail. And so I just kind of penned the term individualized geriatric mouse medicine. And um, that's kind of a model I think that we can use going forward. But we just have one prototype drug cocktail. Um, and so I wanted to show this again because um, there's, you know, a vast opportunity to look at drug combinations of, you know, just this short list of repurposed drugs. Um, and, again, Nir Barzilai and his group at Albert Einstein um, scored these depending on, these different parameters. Um, and the SGLT2 inhibitors were right at the top, metformin, bisphosphonates. Um, and here's acarbose rapamycin. And then we're not sure about phenylbutyrate. Phenylbutyrate, by the way, is urea cycle disorder, is used to treat urea cycle disorders in kids. Um, so again, all these drugs are FDA approved. Um, and so how do we really test these drug combinations and what combination do we want to use? Um, so um, one aspect is asking the question, are mice is the mouse model really the model to do all this heavy lifting? No, because it takes 3 years to do a lifespan study in a mouse, um, and it's expensive to do, all these different drug combinations and all the cohorts that are needed. Um, so, we considered some type of invertebrate model. Uh, there's worms, there's flies. People, work with those. They have, advantages. They can be genetically manipulated, look at mechanistic aspects. Um, we decided to go with another model, a really an untested model, and that was the domestic house cricket. Um, uh, you know, a number of reasons. One is I had worked with this a number of years ago, a long time ago, when I didn't have any grant money, and we did some tests, so I was kind of familiar with them. But they have a lot of advantages, um, in at least in terms of drug screening. They have a heterogeneous background, at least the crickets we're using. Um, they don't have any larval stages. They just go from nymph to juvenile, mature adult, middle-aged, geriatric. And you can see how this compares to the mouse lifespan and the human lifespan. Uh, so they also, um, have can eat the same diet as people. Um, we feed them rodent chow. Really, really easy. They're easy to maintain. Um, and so let's look at some data here. Well, first, I just want to give um, a brief look at the anatomy. So it's very rudimentary, as you might expect, but it's quite well compartmentalized. It has a fairly well developed GI system with salivary glands, and the heart is really part of this dorsal artery, and it gives that dorsal artery a pumping system. Yeah, and there are kidneys. The and This is kind of a simplified spinal cord, although um, you know there are no bones in a cricket. Obviously, there's an exoskeleton, um, and um, um, so um, that will just kind of give you some idea of what we're dealing with. They can easily be um, determined by sex, really, really easy. And they have a short lifespan. We can do a lifespan study in 6 to 8 weeks. So, um, okay. Um, but are they responsive to the drugs that we just talked about? Well, absolutely. Um, you can see here, this is a control curve. Um, and these are large numbers. We wouldn't necessarily do this large number with mice. So we can do large numbers. We can get um, you know really up to speed in terms of using the cohort numbers that are needed for these types of studies. Um, so does it does the cricket respond to performance tests? We can do most of the same things um, in crickets that we do in mice. We've developed a Y maze, which shows that um, you know rapamycin is responsive to increased memory. It's a olfactory-driven memory response, and the acarbose has no effect. Phenylbutyrate effective only in males. Um, we use an open field test using Easy Tracker, an open source program, and simply a cell phone camera for doing the tracking. So we can track lots of different things: distance traveled, mobility, and alertness. And you can see that rapamycin has an effect in males, um, but acarbose and phenylbutyrate have no effect. Same in mobility, but phenylbutyrate has effect in males and so on. Yeah, so yes, you know, it to us it looked like yes, the cricket is going to be responsive to drugs that have already been shown to have positive effects in other animal species. Um, so I had to show this because there are age-related lesions, similar but not the same as in um, vertebrate species. You can see that this is salivary gland here. There's glandular degeneration here. Um, I had to mention the fat body. I didn't point out any liver in that cartoon I showed, but in fact there are um, 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 there's an organ called the fat body. It really simulates some of the functions of the liver, um, and with increasing age, there's atrophy, and here is a part of the brain that shows the deposit of lipofuscin, which used to be considered non, you know, not important, but now it's considered to be significant and associated with several aging-related conditions. Um, so then, where where is our overall strategy? Uh, so for really testing these therapeutic drugs, and I'm talking about combinations. So obviously we need to formulate these drug combinations. So that's up front. That's got to be done. And there are a number of bioanalytic approaches for doing this, as well as just arbitrarily seeing maybe what might match up. And then we do pre-screening. in the cricket model, using um, you know various combinations, probably starting out with drug pairs. Um, and then we can do pharmacokinetics in the cricket, and then we move to whatever is promising to the mouse health span, not lifespan, health span. Um, and then from there, it's a little murky right now. Where do we go from the mouse? Do we use human organoids and or organ-sized cultures? Not really well developed, but I think there's a lot of research going on for this. I mentioned pet dogs and cats. Um, as May knows, there's a, a dog aging program here, centered here, uh, and we're working on maybe getting a cat aging program started. But we don't know where that might be going. Um, and so here we are, with clinical studies. But the real um, aspect of this is to have a large number of different combinations that may be effective for different situations. Remember, we were talking about individualized geriatric medicine. So, what combination might fit one may not fit somebody else, or maybe even one drug. Uh, and this is probably going to be always in combination with um, lifestyle changes. Um, so, um, that's sort of the end of the data I'll be presenting. And it looks like I'm pretty much on time here. I wanted to just mention geroscience training. Um, yeah, everybody's kind of probably heard, about geroscience, um, but um, maybe they don't really understand or appreciate the significance of geroscience. Um, so several years ago, we we wanted to start people early at the undergraduate level. We started the geroscience undergraduate research training program. We call it GERT. Um, and we used to promote this a little bit. We don't even promote it anymore. It's we don't have we don't have enough slots for the people that want to get in this program. We can take 6 to 8 students per year. It goes from fall quarter, starts in fall quarter to spring quarter. Um, and they get credit for this. It's not a formalized course yet. Um, we just have them sign up for special projects. Um, but we're thinking about having this as a organized course. Each student has a mentor and works on a research project. And the objective, the expectation, I should say, is to present their findings at the Mary Grace Undergraduate Research Symposium in the spring. So I did want to mention that. And I know there's increasing interest in geroscience training, both at the graduate level and even at the geriatric fellow level. Um, so um, I wanted to mention this because um, we have started this program, and um, students seem to really like it. Of course, we get really highly motivated students that want to do a good job. Okay, um, so um, I just want to make some acknowledgments. And this barcode is for anyone that wants to evaluate this presentation. It would be helpful to get some feedback. Um, and I'll stop there for discussion. And it looks like we have about 10 minutes. So
Thank you so much. And thank you for telling